let's get going. So welcome to the first Ebba Biotech webinar of 2023. We are a Swedish company. We're founded in 2015 based on interdisciplinary collaborations, and I guess that's what a lot of you are interested in today, and that's what we're going to hear about today. Today we're going to focus on AmyTracker, which the focus of that will be looking at amyloids and other protein aggregates. And to talk about that today, I think it's fair to say we have got one of the best people that could talk about that. In fact, no, not one of the best people, Per. It's the best person, especially with your link to Ebba, which we'll go into as we see the presentation. But we're here today with Professor Per Hammarström. He's a professor at Linköping University, and his lab focuses on protein misfolding. This is what they're experts in, looking at amyloidosis, prion disease, and they use lots and lots of different techniques to basically understand these very, very important global diseases. And Per has been with Ebba from the very beginning, very, very important, and it is a huge pleasure to welcome him to the stage today. And without further ado, I'd like to hand over the floor to you. Per, welcome.
Thank you so much, Ben, and thank you for the very nice introduction, and to Ebba for inviting me to this seminar. So what I will talk about today is amyloid fibril polymorphism in proteinopathies, which is diseases caused by protein misfolding. And of course, amyloidosis is one of major proteinopathies there are. But before going further, I also want to make that disclosure that Ben was referring to. I am a very minor economical interest, but they are there within the Ebba Biotech company commercializing what we call LCOs. Here, Ebba calls them AmyTracker, sorry. So to the scientific topic, amyloid diseases are caused by proteins that misfold and accumulate in different organs. All these diseases are progressive and degenerative, so they will grow worse over time, and it can take a long time before you actually show any symptoms. They occur as both systemic and localized diseases, meaning that they can be localized to one particular cell type, or they can be all over the body. And they're all caused by aggregation of proteins, and they are quite specific and defined. One particular protein aggregates in a specific disease. And this can happen in many, many organs. It can happen systemically. For example, for TTR amyloidosis, can affect peripheral nerves, the heart, and the brain. We also have proteins, which we call them localized amyloidosis, which in many instances are localized only in the central nervous system, in particular the A-beta peptide that we will focus on today. It's also interesting that these diseases are self-perpetuating. What I mean with that is that once aggregated, misfolded protein accumulate as amyloid fibrils, they are self-catalyzing. So as long as there is monomeric building material, substrate for that particular fibril type, it will continue to accumulate in a process which many believe, and we believe, is caused by seeding, that the fibers fragment and self-perpetuate. What defines these diseases once these proteins are initiated and progress is that the control systems that we usually have in all our cells, and that's why we're healthy and you know, have a functional organ in younger years at least, is that the control system seems to fail. So the unfolded protein response doesn't really cope with what's going on. Hence the molecular chaperones and other protein degradation processes are overloaded, and hence the cells have an issue. Many times it causes mitochondrial dysfunction, leading to oxidative stress. Cells feel bad. This will also trigger inflammatory processes that eventually will lead to cell death and organ failure. So where you see the aggregates accumulating, all these nasty things are going on, and eventually it stops functioning totally, and that's usually a fatal reaction, regardless of a fatal outcome, regardless of the disease. So this is very serious and of course a major health problem for many patients throughout the world. But as scientists, you know, there are also many fascinating aspects about these diseases, which are stemming from an underlying interest in protein structures and how proteins behave. And I have a background in protein folding, and in protein folding, it's known, and this was a Nobel Prize 50 years ago, is that from a gene you express one protein sequence, and this protein sequence folds into one specific three-dimensional structure, which provides that protein with its function. So one fold, one sequence, one function. In misfolding, what we see, and that's what we call conformational polymorphism, is that one sequence gives rise to many different folds, which by extension, of course, suggests that they have many different functions. So different types of amyloid fibers that form will have different activities. And they are, as I remind you again, are self-replicating. So they can sort of be dominated by certain structures. And this is very interesting concept from a protein structure point of view, and it's also, I think, very important for disease progression and for diagnosis of these diseases. So what we will focus on today in my examples, that's one of the major projects that we work on in the lab, is to look at amyloid beta, which is a protein associated with Alzheimer's disease. It's one of the sort of most known and canonical amyloid proteins in the field, and one of the most highly studied proteins, I think, in the world. What's interesting with A beta are, of course, its connection to the biggest amyloid disease there is, which is Alzheimer's disease, but it's also that it's very highly polymorphic, and that's been shown from many labs and many beautiful studies over the past 30 years. And one of these very obvious polymorphic examples are the structures that have come out from many labs, from solid state NMR spectroscopy, from cryo-electron microscopy, is that this is a cross-section or a pi-mole representation of cross-sections of structures of amyloid beta fibers, both amyloid beta 42, which is the most aggregation-prone sequence, and A beta 40, which is the most prevalent isoform that we have in the brain. But as you can see from these cross-sections, they're quite variable. In one structure here, you have one of the monomers are folded in this manner, with hiding the C-terminal alanine here. But other structures have a completely different folded monomer, and they have exposure of the C-terminal alanine, showing that they are vastly different. You also see vastly different structures or structure models for A beta 40. So it's not only A beta 42. And these structures all originate from fibers that have been fibrillated under careful conditions in vitro, and then the structures have been determined by different methods. Very recently, Sorcheres and Michel Godert have purified A beta fibers from patients from Alzheimer's disease. So these are fibers formed, or structures of fibers formed inside the brain. And also there they have evidence for polymorphism. So at least two major types of amyloid fibers have been found in humans, and I would postulate, and I will show some data that suggests that there are quite many more amyloid polymorphs in human brains. And this is, of course, quite interesting. And what we see, we'll take an amyloid beta slurry of fibers that we formed in the test tube. You throw it onto an electron microscopy grid, and you look at it with electron microscopy, you will see that they have quite different structures. So here you have an example of a bundle of fibers, which are at least 5 filaments packed together, and they have a very long or wide twist in that fibril type. But in proximity, you see from the scale bar, it's only a few hundred nanometers away. There are two other fibril types, which have two filaments, and one has a tight twist and one has a long twist. And then they sort of merge. So obviously, the amyloid polymorphism that we have from the folded structure can also propagate into more large scale structures. And so on top of the folding polymorphism, that will be the fold of the monomer that we looked at before, we also will have an assembly state that can propagate. And this can be quite easily demonstrated. If this would be a monomer, the cross-section of a monomer, such as the ones we looked at, we assemble that into one single filament, having the monomers stacked together. We can take these filament and pack them face to face or back to back, and they will have completely different structures. One will have a tight twist, one will have a longer twist, just in this very simple experiment in the computer. You can also take 3 of these and stack them together into a trimer, which looks like a Toblerone chocolate. And that will, of course, render a completely different polymorph. So same building block can also build different structures. So the complexity here is quite dramatic, I would say. And what's also highlighted here in red are the interfaces or the growth ends of the fibrils. They will have quite different chemistry, even though the constituent monomer is identical. So with this said, knowing the protein structure determines their function, it's quite obvious that you can have a quite dramatic influence on nasty things if these proteins accumulate. And which type is the most toxic, nobody knows yet. But that's one of the things that we're trying to understand. And we want to look at this not only in the test tube that you have to the left here, but we want to look in this, of course, in the human brain. And also there are other organisms that we use, the transgenic fly or the transgenic mouse. And of course, here you can get high resolution structures, high resolution information, but the most important for disease is on this end. Then you lose the resolution, but it's more biologically relevant. And here you're sort of in between in these experimental animals. So how can we translate this in vivo situation towards the high resolution in vitro situation and vice versa? And there we have over quite many years used fluorescent amyloid probes as our favorite type of method, because that can be applied both here, 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 and here, and what have you, and look at the same thing if the fluorescent amyloid probes are specific for the amyloids you want to look at. And one of our absolutely favorite type of probes are the luminescent conjugated oligothiophenes that has been spearheaded, the development of those has been spearheaded by Peter Nilsson's group. So we also call them the Nilsson probes. And they're all composed of repetitive monomers of thiophene monomer units that can be assembled in different lengths. They can be 5-mers, hexamers, heptamers, they can be tetramers, and different variants of these. And over the years, I'm not sure if this list is complete. I've added a few more proteins since I did this presentation last time. Is that we can look at neurodegenerative amyloid proteins, we can look at systemic amyloid proteins, localized amyloids, and also amyloids that originate from other systems, synthetic or from viruses or bacteria. That all are positive with LCOs, so they can be detectable. But on top of that, we've also, in many of these instances, shown by the virtue of these molecules that they are polymorphic. And we interpret this polymorphism from the spectral output of the molecules. So if they vary a lot and in a systematic way, we can see that they are different, even though they're staying in the same protein. And Peter has really made huge contributions to many groups over the years, including our collaborative work. And it was, by coincidence, there's actually a nice article about Peter Nilsson's work and about his research group in this week's sort of news highlights at the Linköping University website. So I encourage you It's also in English and in Swedish. Encourage you to go to the Linköping University website, and you can read about Peter's research and new probes that they are developing, which are quite interesting for exactly the stuff that I'm talking about today. So one of the common questions that we've got over the years and is, okay, the LCOs, what do they do? Where do they bind? How do they bind? And I would say, there's nothing magical about these probes in the sense that they are different from what is used by many other groups. They bind to, in many instances, the same sites, especially the Congo red binding sites. In some instances, they have new binding sites, which is, of course, interesting in their own respect, but it's not magic in that way. They don't detect anything else. They detect amyloid fibrils, such as Congo red, which is used, since 100 years in pathology. And the first high-resolution model structure of Congo red binding to an amyloid fiber was published 10 years ago or so by Adriano Aguzzi and Beat Meyer, working on HET-S, which is a monomorphic type of fiber. So it's a great model system, comes from a yeast prion. And Congo red binds in a groove. This will be the growth axis of the fibril. Binds in a groove which is hydrophobic in nature, but it's also outlined by a repetitive ladder of lysine residues, where you compare the sulfonic acid of the Congo red molecule with that lysine. And a few years later, it was shown, also work together with Adriano Aguzzi, that PFTAA, one of these oligothiophins, binds in the same groove as Congo red does, and also pairs with the lysine ladders. But what's interesting about these thiophins, or oligothiophins, compared to Congo red is a very rigid molecule. Thiophins can snake into this binding site and thereby impose a very high affinity binding to these fibrils. They adjust their conformation in relation to the binding site. And that's, of course, a very interesting from a binding specificity and affinity point of view, but it can also be used as a spectral marker that the binding site is different from different fiber types. So that's how you can look at conformational polymorphs. That's how we interpret that, I should say. We haven't completely shown that yet, but that's been our interpretation. And these changes can be quite dramatic, but they can also be quite subtle. So what has been spearheaded by Sophie Nyström in working in my group has been to, use a combination of probes to increase the contrast, looking at conformational polymorphs, so how we interpret the conformational polymorphs, mixing a red molecule, this would be HFTAA, with the blue molecule QFTAA, and thereby see how they stain and which parts of an amyloid plaque that gets stained by these molecules. And the example here is that we have a fairly young mouse or a middle-aged mouse at 12 months of age, staining an amyloid plaque, and looking in the spectra here, you see a heat map. This will be the spectra. You only see essentially fluorescence from the HFTAA molecule all over the plaque. It's a cross-section here. There is some variations that could be used for interpretation of just one molecule binding. But if you add QFTAA in an older mouse, so this will be 18 months old, you see that the core of the plaque, you see it here by eye, the core of the plaque has a different shade. It's blue compared to the periphery, which is orange. And that's, or red. That's where HFTAA binds. But in the core of the plaque, you see QFTAA binding. It's quite dramatic change in fluorescence spectra here. So something happens to the plaque as the mice age. And this we have interpreted as a maturation of the plaque structure, which is a different polymorphism is appearing at an older age, which can be influenced by age, by genotype, and different things. And one of the sort of molecular evidence for how this is going on is that you can also do the same type of maturation profiling in vitro by pure systems. So just A beta 40 in this case being studied over time, fibers have formed, but there's a different transition occurring after fibers are formed. So in this phase, this will be a ratio of this molecule binding and this molecule binding. So more of this binding, the ratio goes up. That early fibril only binds to this molecule. over time, QFDA starts to bind as well. And with electron microscopy, and with AFM, we found that early fibers were quite thin or solitary, but as the fibers aged or became, incubated for a longer time, we saw more of these multifilamentous thick fibrils that formed, and that's presumably where the QFDA molecule starts to bind, and that's sort of a marker which correlates in vivo compared to what we see in vitro. In human brains, the situation is actually quite interesting. I mean, you can't follow humans over time because if you want to look at a brain section, it has to be a human that has passed away. So Matthias Jucker's lab used this method, the SOFIS method, to use QFDA and HFDA in combination, looking at the cores of plaques, as was done in the mice previously. And this would be a section from the brain. These would be amyloid plaques. Here they are zoomed in. You see the color of this core is quite different, even though it's in proximity in this brain. And calculating many of these ratios of QFDA and HFDA and plotting them in a sort of a Cartesian plot, one can see that in this orange would be sporadic Alzheimer's disease, normal Alzheimer's disease, normal, but the non-familial form of Alzheimer's disease. There's a quite big variation of how the plaque cores fluoresce. Some of them are very fluorescent with HFDAA and not fluorescent with QFDA, and some are very high QFDA fluorescence of the cores compared to other patients. And there are other, if you have a predisposition, a familial mutation in the presenilin gene that cuts out A beta, or in the APP, which is the parent protein for A beta, they cluster in different regions of this sort of Cartesian plot of these spectra, suggesting that their structures are different. And the disease progression of a patient carrying a mutation in the presenilin gene or APP gene is, of course, the age of onset is dramatically earlier, and the disease progression can be quite influenced by familial mutation, suggesting that you can read out polymorphism by this method, and it likely has an influence on disease progression. Another collaboration also on human brains was with Yiri Safar at Case Western in the US. And Yiri was very interested in, can this be looked at from a disease progression point of view using the same method again, staying in different regions of the brain, and looking at sporadic Alzheimer's disease. So no familial cases, but they have very dramatically different disease rates. So this would be rapidly progressing Alzheimer's disease. That's a disease that progresses approximately a year, or slightly more than a year, from onset to death. And slowly progressing Alzheimer's disease would be more of a expected type of Alzheimer's disease from onset to death, 8 to 9 years disease progression. So using the same method, looking at different parts of the brain, plotting the ratio of QFDAA over HFDA fluorescence ratio. High ratio would be what we, from the mice study, you have cartoons on here from Sophie's paper, see that the core of the plaque is blue, right? That would be a mature amyloid. An immature will have a low ratio. And you see it's quite variable here. But what's interesting is that the rapidly progressing Alzheimer's disease seems to have more mature plaques in the temporal lobe, less in the occipital lobe, and quite minor amounts of mature plaques in the frontal lobe. But in the slowly progressing Alzheimer's disease, there are more mature plaques in the frontal lobe, sort of intermediate in the occipital lobe, and less in the temporal lobe, suggesting that the disease progression, we would expect, of course, a mature plaque to be older from the mouse studies. The disease progression will go from the frontal lobe through the occipital lobe towards the temporal lobe in the slowly progressing Alzheimer's disease case, but in the rapidly progressing disease, it would have the inverse path. Purely speculative in one way, but it's very suggestive and very interesting how this can be followed, because these patient groups, of course, are quite different. So it's very suggestive. It can also, of course, have to do with different replicating local environments, can be many, many other things that I will see if I can end with today. So LCOs is definitely our favorite type of molecules that we look at with fluorescence spectroscopy and microscopy. But there are also other classes of molecules that we also have worked on quite a bit over the years. And Congo red is still the preferred molecule in clinical pathology when it comes to looking at tissue sections and defining an amyloidosis. It's still a hallmark of amyloid that's a part of the classification of this being an amyloid aggregate. But Congo red has some disadvantages. So Klunk and Mathis have developed quite a number of derivatives of Congo red that are very interesting in their own right. And I will talk about a few of them that we have looked at and that we have also made our own versions of, showing that amyloid polymorphism by fluorescent molecules can also be detected by other probes than LCOs. So it has nothing, I mean, so it's not an LCO specific type of observation. It's something which has to do with the proteins. Just have to know how to use the probes. And we started with these scaffolds and changed them. So we had a very talented PhD student a few years ago, Yun Shang, who was working on redesigning X34 probes, which are starting out as Congo red analogs. And one of the molecules that Yun developed was this BTD2 molecule. And BTD2 is environmentally dependent. So if it's dependent on the polarity. So if you have an apolar environment, it's a blue shifted molecule. If it's a polar environment, it's red shifted. So taking this molecule, staining amyloid plaques from a mouse brain, you see that the core has a completely different color compared to the periphery of the plaque, or called the corona of the plaque, suggesting that this probe is sensing different chemical environments in the core compared to the periphery or the corona. And this can also be quantified, both the intensity and also looking at the spectra from the core compared to the corona, and verifying the spectral image that we have a different chemical environment. So that the core is different from the corona in an old mouse. This is a 19-month mouse, like the ones I showed from the QFHF studies. We see very similar types of what we interpret as polymorphic differences of the A beta structures. And then going back from brains back into the test tube, what we want to do, it's quite hard to do, but try to do this, and Johan in the lab is really working on making monomorphic amyloid fibers. And monomorphic amyloid fibers is hard to make because they have a polymorphic nature to them. But if you use careful conditions, in this case, it would be a tris buffer making A beta fibers. See, they are quite thick and quite bundled in this case. We can also look at them with this negative stain electron microscopy and cryo-EM microscopy. We expect these fibers to be very similar to fibers that have been published in the past under pH, neutral pH, physiological-like conditions, what we call it in the test tube. Compare these fibrils, their structure, to these fibers that we call the Gremer fibrils. They're made from the protocol from Lothar Gremer. It's an organic solvent and acidified by TFA. These fibrils are long and slender, and they're quite monomorphic. These are also, in a sense, monomorphic, but they have a more of an assembly polymorphism to them. And these fibers are very, very interesting because they're monomorphic and they have a completely different structure. It's very you can replicate this many, many times. It's a very nice protocol. So what Ganesh did working on these fibers is that he used another of the Klunk Mathis molecules called X34 Congo red analog. Staining fibrils made under these conditions and staining fibrils made under the Lothar-Grimmer conditions with X34 and observing them with fluorescence lifetime imaging microscopy, or FLIM, and analyzing this data in many different ways. But what you observe is that the FLIM or the lifetime of X34 when it's bound to these fibrils compared to the Lothar-Grimmer fibrils are distinctly different. And the lifetime, the short lifetimes, appears to be associated with X34 binding to this type of fibril. And there's you can easily separate these. So Ganesh has also looked in brains, going back then to the brains. This will be from human patients, either amyloid plaques or amyloid beta associated with vascular amyloidosis, or CAA, and quantifying the distribution of lifetimes that you find in these plaques or in the CAA. In humans and in mice, you see a very dramatic distribution difference, as we expect from everything else we've looked at, that you have a quite wide distribution of lifetimes if the probe reports on polymorphs. You see that also in mice and humans. In particular, this distribution is quite dramatic. It goes from very short lifetimes to very long lifetimes. But then you can segment this, of course, like we've done with other probes. You can segment this into what's in the core and what's in the corona. But then Ganesh also says, you know, I really do observe something which I call the ring structure. So we have what he calls the corona would be the outer layer. Then we have a ring structure and a core structure, and they have distinctly different lifetimes within the plaque. So Ganesh calls this the kiwi model. You have a core, you have a ring, and then you have a corona, which is quite interesting. And this is from one probe, X34, just observing it by fluorescence lifetime imaging, because the spectra don't change in this particular case. So we find this method very interesting, and we hope to publish it quite soon. So we have, of course, other scientific questions than just looking at, you know, different polymorphs forming. It's very, you know, important to understand why are different structures toxic, and some of them might not be very toxic, even though if you get amyloid or A beta amyloid accumulating in blood vessels, that's certainly not a good thing, because you can have a stroke that can occur from vasculature bursting in your brain. But this is a topic that we're trying to pursue using transgenic flies, which are very, very sensitive to different types of A beta amyloids. We're also very interested in understanding which polymorphs efficiently spread within a human or an animal, and that's an assay that we can do in vivo, and we can do the assay also in vitro, because we're still back to this. We want to look in vitro, in vivo, and try to relate that to what happens in human disease. So Fariana Parvin, PhD student in the group, is working very intensively on this, trying to understand seeding activity and how this relates to structures that form in vivo. One first simple experiment you see up to the top left is only pure synthetic. So in the test tube, taking amyloid seeds of early or late fibrils formed from A beta 40 or A beta 42. We see for A beta 40, which is slowly progressing rate, that the gray here is the unseeded reaction. If you have an early fibril formation reaction, it doesn't really seed, likely because you haven't started to convert the fibrils yet. Late fibrils are very efficient seeds for A beta 40. A beta 40 is seeded with A beta 40. For A beta 42, what you see is that the later fibers that have matured over 24 hours have a less seeding potency compared to fibers that are formed early, 7 hours. You don't even see a lag phase here. So that's an interesting backdrop to what we will look at on the next slide. What also has been done here is to take amyloids prepped from transgenic flies, which express in different cell types, or from different mouse models that express A beta 40, 42 mixtures, or A beta 42 only. Propagate these fibrils and then use those amplified seeds for new reactions. And as you can see, the seeds that form have seeding capacity, but it's not dramatically much better than what you see when you have pure A beta 40 and pure A beta 42 as your substrate. On the other hand, what mice and humans express in their brains is a mixture of A beta 40 and 42, and of course, many other lengths and variants. And this mixture of A beta 40, dominated by A beta 40 and A beta 42, actually don't seed very well with fibers that are formed in vitro. But if the fibers originate from an animal host, like in the Drosophila or from the mouse, have been amplified one step and then being used as seeds, the amplification or the seeding efficiency really goes up quite dramatically. And it's easier to see when we zoom in. Recombinant with recombinant don't seed very much, but if it comes from a fly or if it comes from mice, the original seed seems to be much more efficient. And this is the topic of her thesis project, to understand what makes amyloid fibers in different animals, or different polymorphs that we expect to be found in different animals, different from what you make in vitro, purely in vitro. One of the successful experiments that has been done recently, we've done many seeding studies now, both Fariana and Sophie are seeding mice with fibers from different origins. I just want to show this because it's been very interesting. Pure recombinant fibers then going into a mouse. So instead of taking the mouse as the seed, you take the recombinant fibers going into the mouse. Inject them at an early age, waiting for 6 months, and then before the mice show spontaneous formation of amyloid plaques, you look at the mouse if the seed has imposed amyloid formation in the mouse. A beta 40 is not really a great seed. There are some plaques, but not many. The mixture of A beta 40 and 42 can show some plaques, but it's not all over the place. But these early fibrils formed from A beta 42 by itself at the end of the growth phase shows dramatic seeding capacity. So there are layers here in the hippocampus which are completely full of amyloid plaques. There are vasculature which are full of CAA, and there are plaques all through the parenchyma here. And the plaques are, quite massive, fibrillar, and nice. And the CAA is something which you don't expect in these mice until they're well above 12 months of age. So this has definitely been induced by the recombinant fibrillar seed. And of course, the next step here is then try to use, in this case, the brains are staying with LCOs, HFTAA, is to go back and look at, can also the structures that you make in vitro, or the structures that comes from one host, be replicated in the mouse and a monomorphic plaque. So now Johan's Grimer fibers, for example, is also being injected into the mice and see if we can make less polymorphic aggregates than we expect to be formed in this particular case. So why are amyloids different in humans and mice and probably in flies compared to what you make in vitro? Except for the seeding phenomenon that we know is interesting, is what replicates fastest. One suggestion has come from Marcus Fendrich's group, I call it the Fendrich hypothesis, is that you have actually proteolytic selection going on in vivo. And the proteolytic selection would mean that only the most resistant strain would actually prevail, and hence the fibers that form in vivo will be selected for by being resistant to degradation. There could also be other factors that can influence polymorphs. And one thing that Johan Larsson in the group is looking at when he's not making amyloid fibers is his favorite project is working with molecular chaperones, and he just published, I think, a very interesting study on molecular chaperones. It's a pure in vitro experiment, but it's quite surprising results. First result is not surprising. He take human HSP10, which is a small chaperonin co-chaperone, and having quite high concentrations, or still substoichiometric, but you have high concentrations compared to what we look at, he can slow down fiber formation. The blue would be the absence of chaperones, the orange would be in the presence of 1 micromolar chaperone. It slows down conversion. You can see the T-halves here. If you increase chaperone concentration, you slow down the conversion time. What was and that's the expected effect of being a molecular chaperone. What's interesting is that when he looked at very low concentrations of chaperone, it seems to be a more rapid fiber conversion rate that happens. So he took he focused on this area of very low concentration, in the tens of nanomolar range of chaperone. Compare that to seeding. So this is now on a different x-axis, as you see, because we're focusing on the fast happenings of this reaction. The blue is still without any seeds or chaperones. Converts with a half-time of approximately 5 hours, as it does here. And then adding seeds, that would be the black curve. It's faster as expected, you shorten the lag time. But interestingly, if you work with 1 nanomolar, 5 nanomolar, 10 nanomolar of HSP10, it speeds up approaching self-seeding reaction of Aβ42, suggesting that the chaperone is actually steering this fibrillation pathway towards a faster fiber formation. And Johan has some data to suggest that it actually also influences the structure of the final form fibril. And our model is that these chaperones, which have mobile loops, binds to fibril ends. This will be a prion, but this is a Aβ fibril, and thereby can stabilize initially form seed nuclei or niduses that can then self-propagate. But if you have too much of these fibrils or these chaperones, they will cap the fibril ends and hence prevent growth of the fibrils, and thereby they will, at high concentrations, they will slow down fiber formation, and that's hopefully what they're doing in our cells instead of speeding it up. So with everything I've said, I have a summary slide here. I hope that I have convinced you that fluorescence probe profiling is a valid way of using sort of translation or a Rosetta Stone for converting spectra, intensity, lifetimes of fluorescent molecules into interpretation that amyloid fibers can be polymorphic or monomorphic. And that amyloid fibers, shown by us and by, of course, many groups now, that amyloid fibers show this vast conformational polymorphism on many levels, not only how the fold of the monomer is, but actually how they stack together. And our hypothesis is that if you have a very stable, densely packed fibril, they will be less neurotoxic. That's been, I haven't talked too much about this now, but they also will have less effects of spreading because they will be more inert. And likely have less seeding capacity compared to early fibrils that will have more active surface area and maybe other structures. And that this in vivo concept of how the polymorphs propagate are very interesting and probably a key to get the correct diagnostics, especially when we use PET ligands that's dependent on the structure of your target, and also to be able to treat amyloid diseases, including Alzheimer's disease. So with that said, I, of course, want to thank everyone involved. This is people in my group. This is Ganesh that I mentioned now, Ganesh. I mentioned Johan, I mentioned Faryana, I mentioned Sophie. I also want to mention, of course, Peter. I've shown a picture of him already. And then collaborators, Peter Konradsson, Stefan Thor, Walker Jackson, and so on and so forth. And I want to thank my funders for the work we do in the group, and thank you guys for listening.